Thank you, Svetlana, for the kind introduction. I didn't expect you to read all those things. That's why I put them on the slide, right? It's too long. Okay, so this is before I start with the actual slides. I wanted to mention, first of all, I'm delighted to be back, and it's kind of emotional. I see many faces, friends, and we work too much sometimes. I feel like we see each other too. But when I was here, usually when I gave a talk, I think I gave 1 or 2 talks, was about modeling cancer evolution. That was how I came to this field, and thanks to Giovanni, he expanded my horizons, introducing me to sequencing data and all the good stuff. And then I believe I gave a talk during COVID, which was more on cancer etiology, or at least that was the main focus of that talk. And so I thought today I would do what has become probably what takes the majority of my time today, which is focusing on monitoring methods for cancer early detection, as well as risk prediction. And in fact, even though the title starts with risk prediction, then early detection, and then MRD, I'll go in reverse, okay, in the presentation. And I'll keep the last, which I think is the most exciting, at least for me, part. So, some disclosures. Okay, so let's start with MRD, and I hope to give you. I was trying to think what is that makes sense to give as a presentation here at Harvard. Great statisticians, computational biologists, and I hope I made the right choice, and I just want to focus on some ideas more than any type of detail. Okay, so I'm not going to present one study in great detail, but hopefully you will like this. And so we all know that thinking about monitoring patients, right? You can monitor patients even before surgery, and of course right after surgery to determine what is the residual disease in that patient, as well as you can survey using blood testing, for example, on how the treatment is going, right? You could use the technologies that are currently being developed for, say, switching therapy if a patient is not responding anymore to the current treatment. And then, of course, as I said, you can check for relapses and so on. So in all these phases, there is a lot of technology that you can use and that is being developed, has been developed, and is being currently developed. And this is a very fast-moving field. So here is just my little example on something that I worked on. It's called the dynamic study. And this is a 2018 paper, but actually the first one is in 2016. And essentially, we started by doing some observational studies. This was led by Dr. Bogenstein lab in collaboration with Peter Gibbs in Australia. And there, as you can see, what is important to, the only thing I want you to notice about the schema here is, the trial is that essentially we were collecting blood before surgery, right? And in fact, even before chemoradiotherapy, and then after a long course of chemoradiotherapy, and then finally after surgery. Okay, so there were 3 time points at which we would collect blood. And then this was just an observational study. So, we would analyze the tumor tissue, of course, to determine what were the specific driver mutations in the patient, and then look for those in the blood through our assay. And meanwhile, the patients were going through standard management, of course. Okay, and what was nice to see, of course, was that while pre-treatment, you couldn't really tell the difference between patients. When using ctDNA, so circulating tumor DNA, you could actually post-chemoradiotherapy and even more post-surgery, you can really tell a difference in terms of, progression-free survival, between those that were ctDNA positive and those that were ctDNA negative. In fact, I think was 13 times. So any ctDNA positive patient were 13 times more likely to recur than those that were negative. So that was nice, and so that was observational, and then that was followed by a larger study and observational. Sorry, this is interventional, so a prospective randomized study where it was about 450 patients, stage 2 colon cancer. The basic question is essentially, these patients have surgery, and standard care would be to give them chemo. And do they really need it or not, right? Can we use blood testing and this type of assays to spare chemotherapy to some patients where really surgery may have been able to remove everything? Right? And given the observational studies I showed before, then we did this respectively. And so there were, of course, 2 arms. In the interventional arm, we do the blood test, and based on the essentially the algorithms that we developed, we will provide a score, and that score will determine whether the patient would undergo chemo or not. And the good news is that we achieved non-inferiority results, but exposing essentially half of the patients to chemo that would have gotten, when compared to the control arm. And so obviously these are very exciting approaches. Very good news for patients, right? The ability to avoid exposure to chemo. Okay. Initially, I thought I would give you some sense of what we did, because actually statistically there is something interesting here in this study. But then I decided not to, because this is actually a perfect example of something where you work for like 2 years, you come up with something that you think it's really cool, and maybe it is, or at least decently cool. And then meanwhile, in the last 6 months, while publishing this paper, Augustine colleagues, and I just did some calculation for them, come up with a new assay that completely destroys the previous one in terms of performance. And so as you can see on the right side, okay, the previous was called SafeSeq. This is SaferSeq, and essentially on the 3 panels on the left show, the performance of SafeSeq, and here is SaferSeq, so the new approach. And as you can see, essentially noise is completely gone, okay? And the basic idea is just that once you introduce the ability to check both strands, so both Watson and Crick for the mutation, okay, now the noise is essentially reduced to 0. And so you need very little statistics at that point, actually. Okay, so I thought this was an interesting example of, how you can work on a method, and can be somewhat complicated too. But technology is developing so fast that that it's always ahead of you. That's at least how I feel in general. And in fact, I claim that for people working in our field, it's very important to not just be user of technology, but if possible to interact with the technology. So that's what I've been trying to do. Okay, so that was it for monitoring. Now moving to cancer early detection. Well, let me first just mention, I know everyone knows this, but there is an enormous cost in terms of, first of all, lives, so survival, right? Here you see some data. The ballpark is what a woman with breast cancer in stage 0-1, 99% probability to be alive 5 years later, versus if it's like in stage 4, that same person would have a 25% or so survival, at 5 years. And that's for breast cancer, but that's for many, many cancer types. There are a few exceptions, of course. Pancreatic cancer is a bit different, for example, but that's the general picture. And on top of the survival, which is the most important aspect, there is also a financial aspect, right? So the cost of treating a patient in stage 4 is very different from treating a patient in stage 0-1. And we all pay for that. It's a cost for everyone. And so what is very surprising is that, as of today, if we're talking about general screening available, I'm not talking about tests that you can buy if you have the money to spend $1,000. There is no screen available for many cancer types. Okay, here I listed few, so ovarian, liver, pancreatic, esophageal, stomach, and I think it's unbelievable. This is very personal. Like, my father was diagnosed with pancreatic cancer in December, and in January he passed away. So, how is possible in 2024 that this is where we are? It seems incredible. And so, anyway, so I think that to me, working on early detection of cancer is one of the most exciting things that you can do in cancer research, for sure. And so it's not just that we don't have screening for many cancer types. Also, for the ones we have, there are still problems. The uptake is far from ideal, and on the right you see some of the numbers, right? And one of the reasons is that some of this testing is not very pleasant, or it's not as simple as what I think is a blood test. I think it's simple. But if you have done a colonoscopy, I think you know what I'm talking about. Okay, so and so I'm delighted that at the national level, prevention of cancer and cancer detection have now really taken the front stage. It's of the 8 goals, are the first 2 are prevention and early detection. And so just let me put this here just to say that I was at Johns Hopkins, and everything was going very well. There was absolutely no problem, and I really enjoyed what I was doing there. But as City of Hope came an opportunity where the cancer center wanted to invest on prevention and detection, opening a center, and so that was a very exciting opportunity. And what I like particularly is the combination of, on one side, a technological branch, which is this Translational Genomic Research Institute in Phoenix, with a clinical network, the City of Hope has, which has become very large actually now, 5 cancer centers across the country. And so we have, work in progress in many directions across many different types of biomarkers and technologies. Okay, so talking about early detection, I'll talk about 2 ways that you can go about it. One is circulating tumor DNA. So you're looking for DNA that comes specifically from the cancer, versus cell-free DNA. Okay, that's just circulating DNA. And I'll start with circulating tumor DNA. So we know the ctDNA sheds, we can find in the blood. Of course, we also know that it sheds at different rates, depending on the cancer type, okay, the tissue type. And so some tissues shed much more than others. So, colon is a shedder, and so the thyroid or glioma, right? Glioma is the one that sheds the least, probably because of the blood-brain barrier. So that's a challenge. But even with that, as you can see, I would say overall, tissues and cancers end up shedding, and so that enables us to develop technologies and approaches to detect cancer early. So this was a case control study where we showed that for 8 major cancer types we could detect with, I think the median sensitivity was 70%, 99 specificity, and of course with performances that vary depending on the stage of the cancer type and the cancer itself. And that was an approach that used ctDNA specifically a panel of about 60 genes, and so well-known driver mutations combined with proteins, panel of 8 proteins. And that was followed, so there was a case control study that was followed prospectively by a larger study on 10,000 women, where we applied cancer-seq, and if a person was positive on the blood test, the follow-up was to do a CT scan. And what the study showed, even though it was far from perfect in terms of results, but what the study showed is that thanks to the blood test, the number of cancers detected was about doubled, okay, with respect to the standard care. Okay, so that's circulating tumor DNA and my experience with that. And then there is cell-free DNA, okay? And I want to spend some time here because I think this is very promising and exciting direction. So cell-free DNA, one thing that I find exciting about that is that it allows you to look for many biomarkers at once, okay? So in cell-free DNA, you can look for fragmentomics. And so I'm sure you're all familiar, but I'll just briefly say, right? The idea that because of nucleosome positioning getting messed up in cancer, the fragment size of DNA changes. And so you can detect changes in the distribution of the fragment length of DNA when comparing cancer patients to normals. So that's a very promising direction. Of course, aneuploidy and breakages, breakpoints, as well as even methylation. We are currently working on adding methylation to our assay. Okay, so that's the advantage. One of the advantages, at least, of cell-free DNA. And I wrote here, it's a more pervasive signal in the following sense. To detect cancer, it's a very hard game, right? Now, if you are trying to detect cancer in a patient through ctDNA, you are literally looking for, say, one base mutated, right? And that fragment coming from that very cancer in the blood of a patient, okay? So it's at one position across the whole genome versus fragmentation patterns and changes in it, as well as aneuploidy, are a more pervasive signal. So to me, it's a bit of an easier game, okay? More than a bit. The first version of it is called RealSeq, it's a technology, which is an amplicon-based approach. So with an amplicon-based approach, what you do is instead of sequencing the whole genome, essentially you can pick your regions, okay? And on those regions, though, you go to much greater depth, okay? So now this becomes a mathematical question. Actually, it's not difficult to answer, but which is it better to see everything in a shallow way, right? Or in certain regions with greater depth.
Still fragment length.
Right, and still doing fragment length and aneuploidy. And in fact, as I said, you can even do methylation with this. And the answer is actually, well, it depends, right? It depends, but I think in general, at least if you're looking at fragment length, this is one example. I hope you can see with the colors, but in blue, you have the amplicon-based approach. In red, you have the shallow sequencing approach. And what you can see there is that, solid line is cancer, dashed line is healthy. What you can see is that there is, I think you can see by eye, there is more separation, basically, provided by the amplicon-based approach. So that's why we chose to go in that direction. And so the original paper, the first one is this one, and Cristian first author, he's a postdoc with Bert. And this essentially, the idea of RealSeq is to use a single primer pair. Okay, so this is a primer pair, just one, and it attaches to the genome in about 350,000 places. Okay, so you get about 350,000 applicants. And the way RealSeq was built was unimodal, okay, in terms of fragment length. And so that's not the actual picture, it's just a depiction of the idea, right? But essentially, you want applicants that have the same, the amplify fragments of the same size. So the applicants should be of the same size, because it essentially the logic was it allows you to make comparisons more easily. If you know the metric they use is based on something, an applicant that essentially has always the same size. And so that's RealSeq, and it looks for essentially, it enriches for ALU elements, which are these jumping genes, and they are all over the genome. And so another aspect of the amplicon-based assay that's important to mention is that it requires very little plasma. So we regularly perform, in fact, usually we can do it with 0.5 ml plasma. But let's say 1 ml plasma is typically what we require. And yes, and as I said, allows us to analyze both fragmentomics and aneuploidy. Okay. So, and I already explained the nucleosome and why that is associated to changes in fragment. Okay, I want to, before showing the results, I just want to show you the algorithm attached to it. So what we do in a particular study I'll show in a second is something called signal. It's essentially it's NMF. Okay, and so we have a matrix of columns are samples and the rows are the applicants. And so in each, in each box, each cell, you have the normalized applicant count. So you are essentially counting how many times an applicant was amplified, and how many reads you have from that applicant. And then there are, NMF essentially gives you two matrices out, right? And one is a matrix of signatures. This is, if you're familiar with the mutational signature work, I think you will find this relatively intuitive. But essentially, you get the matrix of signatures where, signature for short fragments or long fragments. These are just probability distributions, okay, on all the applicants. And then another matrix that essentially provides you the intensities of each signature in each cell. Okay, so it tells you how intense was a particular signature in that particular patient. So that's the basic idea of signal. Okay, so I want to spend a second on this because I think this is actually striking, very important. It's submitted, so it's not published yet. But here is what we did. By the way, this is I hope this is of interest even for those that work in the monitoring space. Okay, so let's think in 1 second. Forget the slide for 1 second. Just let's think 1 second about advanced adenomas. So advanced adenomas are polyps, right? They are dangerous. Okay, this is not just your general polyp, and but they are not cancer. So when you think about finding an advanced adenoma through a blood test, this is a harder game than finding colorectal cancer, right? So what we did at the beginning to see if we could detect advanced adenomas was we used basically the most sensitive assay we had for ctDNA. Okay, so looking specifically for mutations in the blood of those patients. How? We would take out the advanced adenoma from the patient. Okay, this is, I say we, but it was Rocky showing in Pittsburgh. So take out the advanced adenoma, sequence it, just bulk sequencing, find the driver mutations. Patients had between 1 to 5 driver mutations, and then build the panel, the assay specifically, so bespoke assay for that patient, and look in the blood with the most sensitive assay we had at that time for the signal. Okay, now that's something that it's cheating, because if I want to develop a blood test that finds advanced adenomas, I don't have the advanced adenoma at my disposal to sequence and find the drivers, right? But we did have to understand what was kind of like an upper bound, how high of a sensitivity we could possibly reach, okay? So this was just to understand the problem. So when we did that, I don't have actually the numbers. Yes, they are here. Only 8% of advanced adenomas were found in that way. Okay. So then, just because, what the heck, we said, let's do the amplicon-based approach and see what happens. And so what I just described to you, so this real seq with signal attached to that, we just with aneuploidy, we were able to find 40%. So 40% sensitivity, basically. 94% specificity. By the way, here specificity doesn't have to be like 99% to having cancer. Okay, it's okay to be a bit lower. But the bottom line here, and I think what is striking, at least to me, the first time that I saw these results, is that if you ask me before doing this work, okay, we're going to give you a harder problem. It's not cancer, it's pre-cancer. Okay, and we allow you to take out this pre-cancer thing and sequence it and find the drivers and then test it in the blood with the best assay you have, okay, versus an approach that is, you don't even look at the advanced adenoma. So it's, tissue informed against tissue agnostic, right? And you're just going to look at fragmentomics and aneuploidy patterns. I would have said for sure the ctDNA approach would have won. And instead of CFA, the cell-free DNA is by far the winner. And so, again, this brings me back to what I was mentioning before, which is, I believe now I'm even more convinced that the signal of fragmentation patterns, aneuploidy, methylation, because they are more pervasive, they are easier to be detected. And not just that, the one question was, but could be that those are signals that come later in the game, right? If I want to detect something that's even pre-cancer, this is a very early signal. Okay, so maybe some of these things don't get messed up until later, right? What this says is actually that doesn't seem to be the case. Those are things that are actually already altered from the very beginning, at least in our experience. Now, let me say, this is a very small study, okay? You saw the numbers. We submitted it. In fact, from the first journal, we got rejected because it was too small of a sample size. But, and so it was, only 72 samples, but it was a blinded test. Okay, so I hope you found that interesting. Maybe also let me mention, when we think about cell-free DNA, the question is, where is that coming from? And I think that's a very important question. On top of it, we know, as this figure shows, we know that cancer patients have more or tend to have more cell-free DNA than healthy individuals, okay? And so why is that, first of all? In fact, I could use a classification for cancer detection. If I were okay with 20% sensitivity, I could just use the amount of cell-free DNA. That would be enough. Okay, it's just that's not good enough. But because patients have more cell-free DNA when they have cancer, or again, 10 distribution. So why is that? Well, if you asked me before the study, I would have said, well, it's because the cancer is shedding, right? You have a cancer. It's not normal to have there, and this cancer is growing like crazy and producing a lot of, dead cells, and so that's why you have more cell-free DNA. But the answer is that's not really the main cause, main source of the excess, because what we showed last year in this paper is that very recently is that the majority, the great majority of the cell-free DNA, the excess cell-free DNA, is actually coming from white blood cells. So these are cells responding, attacking the cancer, okay? And that's very interesting to me. Also because if you remember what I just mentioned before, which is that Even advanced adenomas, we find the signals so early. Okay, this means that even at that stage, you have essentially a reaction by the immune system, right? Now, the question could be, the criticism could be, well, how do you stay specific, basically? What if someone is having a major immune system reaction, but has no cancer, right? And those are the things that we need to control for, of course. But anyway,
Cristian, this is amazing. I didn't read this paper. Can you describe in 15 seconds how you found this out?
Yeah, basically the bottom line is we looked, and again, this is work with Matt Buttocks and Wibbert, and this is essentially we use methylation, okay, to distinguish different sources of work. Yeah, that's how we do it. And okay, so just to conclude on this part, this is the most recent paper that we published on YASIC just 2 months ago, I think. And again, just applying this technology, we get about 50% sensitivity and essentially 99% specificity. And this is just looking at fermentomyces aneuploidy, but as I mentioned before, there are more things that you can do with cell-free DNA. And it's a very cheap assay, it requires very little blood. Okay. All right. To conclude, we have developed now, City of Hope, we developed something that we think performs better. The B and the best actually was not my naming, but the B is for bimodal. Turns out that when you look at it mathematically, that if you want to optimize, the approach, it's much better to have 2 fragment length sizes than 1. Okay, if you want to distinguish fragment lengths, and so we have a new essentially assay that has a bimodal distribution in terms of fragment lengths. And so we look at, just for the fragmentomics part, we look at fragment length, we look at determining what the protected regions are. So these are regions where there are no fragmentation events. Imagine areas where bombs cannot fall, and motifs, right? So the study of what are the letters, what are the bases, the beginning and at the end of these fragments, and those also can tell us a lot about changes with respect to between cancer and non. Okay, so I can't say more about this for now. So I'll now finish with the last part, talking about risk prediction. So let me actually, before mentioning the next slide, let me just say, to me, early detection, it's a field where there is already so much work and even companies. Okay, it's super exciting, but to me, the next frontier is risk prediction, which is something that Giovanni has been doing for many, many years. And I always thought that risk prediction is by far the most fascinating thing that you could do in cancer research. And if I had to say, I don't know if Giovanni will agree with me, I think he will, but if I had to say, I think risk prediction today is still far from optimal. Okay, it's still very poor. If you ever tried to fill those forms online of some cancer center, it says you want to calculate your risk, and then you start putting like, okay, this is my BMI, this is my age, my gender, and unless you are a heavy smoker or really, really severely obese, in general, the numbers that are spit out are 1.14, 14% more risk, or if that. So I think, why is that? I think the major reason for that, and I know that I may be extreme on this, so this is just my opinion, but from my point of view, a major reason is that we have a pretty, we have some understanding of the germline component of cancer risk. And so obviously, if a woman has a BRCA mutation, that's pretty clear what the increasing risk is and so on. And we also have a decent understanding of exposures and what those, at least the main exposures, like smoking, alcohol exposure, and what that causes in terms of mutational load. We have senators to look at them and so on. But if you, and you don't have to believe these numbers are surely wrong with 100% probability. But if these numbers are even close to the right ballpark in the study, right? And this is just one that's been a series until the most recent 2022, and not just mine at this point. I think there are so many groups, but the Sanger Institute, the Broad Institute. I think everyone agrees today that there is a major component of accumulation of mutations that is endogenous. And whether it's 2/3 or 50%, it doesn't matter. But what that says is that until when we are not able to assess that component, our risk prediction is never going to be very good, because we are basically missing the major player in the game. That's my take. I know it's still somewhat controversial, but and so I'm very interested in approaches that can assess, by the way, not just necessarily this endogenous component, also the others, but they can assess essentially this accumulation, somatic mutation accumulation. And so let me start with this paper, which was done with Giovanni. I think we can claim was the first analysis that used whole exome data to make inference on accumulation in healthy tissues of mutations. Even though we use cancer tissues, and that was very confusing to many people. And so there we observed a significant increase with age that was independent of the cancer. These are all patients with colorectal cancer in this figure. Okay, and control for stage and all, and you clearly see a difference due to age, right? And so this wasn't a final proof. Martin Correa has done since then a lot of work to prove, analyzing tens of thousands of samples, things that we would never be able to do at that time. But here is the proof of concept that if the stage was the same, it really appeared like the age, and so how many years passed before cancer really determined the variation that you said there in terms of in respect to age. And then of course the other thing that's interesting to notice is the large interindividual variation, which the question is how much of that is just noise, just sequencer that are messy, and how much is real. But if a part of that is real, that can be very important, right? So let me just mention one thing to make the point here. So as you can see here, we go from say 50. I believe this was the exam. So yeah, so 50, right? About 50 to patients that say 150. Okay, so if that variation were real, probably it's not. I mean, part of that is sequencing variation, but let's say if that were real, right? That's a factor of 3. So in smoking, we know that smoking causes about 3 to 4 times higher mutational load in the lungs of those that smoke versus a non-smoker. Okay, but that 3, the factor of 3. There is a power law relationship. The factor of 3 becomes more than 20 times higher risk in the lifetime to get lung cancer. Okay, so a variation by a factor of 3 is huge. It's like a person being a heavy smoker not knowing that they do, right? So that was the first paper. Then we published this in 2016, which was, to my knowledge, the next work in this direction. And where again, we showed this age correlation. And these were healthy tissues. These were, we know, cancer, you know. And at the same time, another study came out in Nature, where what they did is they use organoids, okay? Which enables you, once you grow the organoid, then it's very easy. You just do bulk sequencing on it, because the whole organoid should have the signal of the original cell, stem cell, for example, right? And so they also found this age signal. One important thing, so two things I want you to notice, and here again I'll get to be maybe controversial, but so in their analysis, they find, as you can see, about, I don't know, a few thousand mutations in the genome across, you know, in these tissues. And importantly, they find that if you look also on the right, you know, these are very few data points, of course, and the p-value just for whether it's long to see or not, but it's significant or not. But if you look on the right, you can see that the mutational load is not very different across those three tissues. Okay. Any
questions? So I have a couple of questions. So with the risk prediction and understanding of this kind of background mutational load, so is there any hope for it ever to become modifiable? Because, like, well, yeah, you're at higher risk, but it doesn't seem to be immediately modifiable.
Oh, that's a fantastic question, and it's almost a philosophical question. That's why I keep more. You know, I'll give you my opinion. Yes, I think it is, in the sense that, you know, first of all, these things should be able to measure as exposures, right? But let's focus on endogenous component, which is what your question was about. I think there is a baseline background rate, yes, but I think that there is a lot of variation based on what we observe here, and you know, my wild guess is that, for example, you know, the stress, inflammation is just cell death, so new cells, and so more mutations, you know, mechanism could explain some of this. You know, I think, like, you can have the same, you can even have the same environmental insult, and I'm not even talking about smoking. I'm talking about a bad day at work, right, or whatever that is. But because of how we are, or we choose to be, you know, Giovanni with his experience would take the same stressful event much better than, say, I would. Okay, he has seen it enough times to know how to deal with that. So the same external event, stressful event, will cause him to be less stressed, less inflamed, and less mutations. So that's like a modifiable in the sense that I think it's something that where we have even you know, an element of choice, right? We can choose to not become as stressed. At least I think so.
Yeah, that's not a choice usually.
But then it gets to the free will argument, right? Do we have the free will or not, right? But so level of stress. So I would say, to answer your question, you know, just from a purely scientific point of view, I think that I'm convinced that things that can help us with, say, stress management would have an impact on this background accumulation.
Well, I suppose you can think about it as a kind of a quantitative way of taking a disease history, right? Health history.
Right. Yes. And you know, and I mentioned stress just because stress is usually not mentioned, but you know, there are people that think about aspirin, or you know, there may be at some point medication, anti-inflammatory basically medication. That's one direction that I can potentially see.
Yeah, that's a great. Thinking back about your figure that's multimodal, going the next step on the balance function, great question. I mean, you could conceivably be able to identify mechanisms that are behind the increased mutation rate, like mismatch repair deficiency. Example I was mentioning, but there could be others. And if those are the result of loss of heterozygosity in certain important genes like the ones associated with Lynch syndrome, it's conceivable to restore that functionality in cells. So in simple examples, what residual design is asking is the way. Now, that's a simple example because compared to the majority of the rest of humans, if you're Lynch, you're missing a certain function, you get it restored, you're back to square one. Now, the next step is to say, okay, you've shown that this replication effects are really important. You quantify them to a massive proportion, whatever it may be, of the burden of cancer. What if we got rid of those? We would get rid of a lot of the mosaicism in our somatic cell. If there's one person in the world that probably has thought deeply about this, is sitting right in front of me. Do you think that is part of how, from an evolutionary standpoint, we need to be? Multicellular organism, yes, but multicellular and mosaic and with variation in the genome at the single cell level. Or do you think that if we could get rid of the somatic accumulation of somatic variation in our population of cells, we would be the same species without cancer?
Yeah, so it's a great question, very difficult to answer. In a sense, the way I see it is an optimization question. And so I would say it depends on where our species is, right? And at which time point are we looking at it? Because variation is certainly necessary, right, for evolution. So some amount is needed. Your question is, how much is there a right amount? Can we decrease it and maybe even improve evolutionarily or not? And so the answer is, I don't know. But to me, it would make sense that the challenges and the selection processes that were occurring like thousands, hundreds of thousands, millions of years are not the same that we have today. And so maybe maybe those rates and what is optimal changes.
Yeah, but to clarify my question, and it's a completely hypothetical question. What if we could stop the somatic variation without messing with the germline variation?
Or like completely stop it? Well, it depends on who you believe, right? If you're a hardcore evolutionary biologist, you will say that's terrible because we are going to continue to improve thanks to our.
But I'm not stopping the germline variation, so my offspring will be different from me, but my cells will be all identical throughout my life.
Oh, I see. So you're just talking about, yeah, that's if you're just focusing on blocking the somatic, in theory, right?
In what theory?
I'm saying I don't know of any theory that says the somatic variation it's necessarily helpful to us.
So you think it's just a byproduct of the need to have germline variation?
Well, I'm saying we for sure know that is an element that's brought by the genetic variation. I don't know of any work that shows that we need to have the variation. I may be wrong. The place where I can see that being a problem, actually, I'm thinking of my feet, but is everything that has to do, and we are working also on that on immunotherapy stuff, right? So there you definitely. So actually, I take back my answer and say, yeah, certainly you cannot stop it, or at least not for some cells.
You can stop the IG manufactured rush. All right, we're digressing too much. Sorry, I'll stop here because others may have a little bit.
Actually, I have so having mutations is not a synonym of having cancer. And whenever I hear mutation on the law, I was from trouble because if technique is sensitive enough, everybody has multiple mutations. So mutation on law doesn't necessarily mean you have cancer.
Right.
So I think it's where it's important to screen cancer-related mutations for each type of cancer, not just mutational load. It's
Yeah,
not sensitive enough in my mind.
So that's a great point. And I actually, I think you have 2 points. First of all, on the last one, I completely agree. Different tissues accumulate differently. And so ideally, you want to follow each one of them individually. With respect to the, somatic mutations load is no cancer. Of course, I agree. The way I see it is just that, I have, I guess, a probabilistic point of view, which is that, Schetter is parable. So keeping everything else the same, right? Whatever is the microenvironment that you have, whatever is your immune system, capability to fight disease and so on. If I have more mutations, I'm at higher risk. That's all that's how I think about it. So, but yes, mutations are a necessary condition, it's not sufficient, but I believe they scale. I'm not saying linearly necessarily, but they scale with cancer incidence.
Also, some mutations are actually good.
Right, yeah, of course, yeah.
AML, if you have DNMT3A, even if you have better prognosis than other mutations. I also have some trouble with the cell-free DNA. We have heard of cell-free DNA. We have tried so many times. But what is the exact utility of cell-free DNA? I thought, so I don't know. People are talking about these things all the time, but what is the exact position in cancer research?
I know it's a developing field, so it's still messy. The answer is still not fully clear. I think cell-free DNA, as I hope I made the case, has a lot of potential. In fact, I think even for monitoring, I didn't mention that, but if we do so well with detecting advanced adenomas with just cell-free DNA, which is tissue agnostic, how about in a monitoring approach where again we may not have to look at the tissue at all. So the jury's out. We'll see.
Thank you for the talk. Do you have any impression on what are opportunities in looking not at this single time point, the composition of mutation, but longitudinally? Do you have any work in that direction?
No, so that, right now we are working on this technology that hopefully can do a decent job at detecting more mutations that previously detectable. And then, I show you one study would be basically case, we just want to understand individual variation across different tissues. One study that we plan afterwards is with a cohort where, this is a cohort of people that have been followed for 30 years or so. I think follow-up is at least 30 years. And so on those, we would like to apply a bot, say at the earliest time point that we have blood for, and the question there is like, could we have said that there's group, and out of this cohort, there is a proportion pretty large that got cancer. Could we have said based on, the blood 20 years before that this person was going at faster speed than whether it's cancer or heart disease. And then to get to your question, these cohorts have multiple blood collection. And that is very important in terms of, future direction, because, okay, sure, you can tell me what is my speed right now, but if I want to try to, say, become more healthy, less stressed out, whatever it is, stop smoking, I would like to see that I can detect that slowing down, effect in time. So then instead of looking at the number of mutations, you're going to look at slopes, rates, changes in time. So yes, that's no work yet. That is not mine.
Thank you very much.
Thank you.